Julian! I don't know where I felt like I get a little drink around here, do you, bud?
Before one is able to get a drink around here, fermentation must occur to produce the alcohol. Fermentation is defined as the metabolic process that consumes sugar in the absence of oxygen. The products are organic acids, gases, or alcohol. Fermentation is used today in a variety of food industries such as alcoholic drinks and dairy products, and has been a part of a natural process in the human body since the dawn of man. It wasn't until the 17th century that Antony von Leeuwenhoek observed microbials. This is where yeast was first observed, but at this point nothing was known about its contribution to fermentation. It wasn't until 1815 that yeast was proven to be the major contributor to fermenting, when French chemist Joseph Louis Gay-Lussac conducted an experiment with wine making. He first boiled everything that was required for making wine and stored it to ferment, only to discover that nothing had occurred. He then boiled a different batch of wine, but instead of having the yeast in the boil, he let the mixture cool before adding the yeast. This and microscopic discoveries from a French inventor, Charles Cagnard de la Tour, showed that yeast was a single-celled organism which had a major influence on the fermentation process. Their efforts had been proven even further when Louis Pasteur conducted an experiment that showed that living yeast cells turned sugar into ethanol. Pasteur also discovered that there were two types of fermentation when he helped a brewer out with a problem involving batches going awry. His findings showed that the bad batches of alcohol contained a high amount of lactic acid and didn't have yeast cells, but rather consisted of bacteria. He concluded that instead of just alcoholic fermentation, there was also lactic acid fermentation. In 1919, the Prohibition began, which got the people of America on a creative adventure for creating alcohol. The people turned to making alcohol from things that they had around the house, farms, and local stores. They based their alcohol off of malt, cornmeal, sugar, water, and yeast. They would then put their cornmeal into something called a still, which was filled with hot water. Stills were generally made out of a metal that could handle heat, which generally consisted of scrap metal. Afterwards, their other ingredients were left to ferment into what is called a mash. This mash had to be stirred and heated at very specific temperatures and times for the optimal flavor of their drink. The mash is finally boiled and then condensed into a barrel to be able to get their precious alcoholic beverage known as moonshine. Sadly, most people back in the day didn't know the dangers that came with drinking moonshine. Only experienced people that created moonshine knew that the mash couldn't be boiled past a certain temperature in order to get rid of a really bad taste, otherwise known as methanol. Methanol led to some people going into comas or even dying.
The fermentation mechanism begins with glucose, which comes from a variety of sugars. The two main desired products of fermentation are ethanol and lactic acid. This mechanism is for the production of ethanol using yeast as a catalyst for the glucose. The glucose undergoes glycolysis, which produces 2-glyceraldehyde 3-phosphate molecules, which are then converted to pyruvic acid. This can then be deprotonated to pyruvate. NAD+ is used to deprotonate the glyceraldehyde 3-phosphate, producing NADH. This step of the fermentation process also produces 2-adenosine triphosphate, or ATP molecules. ATP can be used as an energy source for cells in the body. The mechanisms for the synthesis of ethanol and lactic acid differ, beginning with pyruvic acid. Here, pyruvate decarboxylase is used to remove the carboxyl group from the pyruvate, which results in carbon dioxide, which is allowed to escape, and acetaldehyde. The acetaldehyde is then protonated by NADH to produce ethanol. This also regenerates the NAD+ used for glycolysis. This process is carried out in anaerobic conditions because air could cause the ethanol to oxidize to ethanoic acid. Fermentation alone is only able to produce approximately 12% ethanol because the environment gets too toxic and kills the yeast. Distilling is required to produce a higher ethanol concentration. There are several important structural features to note in this mechanism. First, glucose is a 6-membered ring with all of its substituents equatorial. Pyruvic acid is an acid because of the carboxylic acid group, and we all know carboxylic acids are acids. Acetaldehyde contains the aldehyde group shown here. Ethanol is an alcohol because of the OH here, and then it gets its name from the ethyl group shown here.
For fermentation, there are two main types of processes, the first being submerged cultivation. Submerged cultivation is the fermentation process by which microbial cells, or yeast, are submerged into the liquid being fermented. Submerged cultivation has three subsets: batch, fed batch, and continuous cultivation. Batch cultivation represents a closed system by which all the ingredients and yeast are added at the beginning of cultivation. The microorganisms and nutrients are left for a set period of time during which the nutrient stock is depleted. The second subset is fed batch cultivation, which is where the nutrients and ingredients can be added gradually during the fermentation process. The third and final subset is continuous cultivation. Continuous cultivation is an open system where ingredients are added and product is removed at a steady rate throughout the process. One of the most common commercial productions that uses submerged cultivation is the process of beer brewing. Beer brewing starts out with the malting of whatever specific barley or grain is used for the desired beer. Those malted grains are then milled and turned into wort through the mashing process. The wort is then combined with ingredients used for flavoring, normally hops, and water in a large vat for the brewing process. The flavored liquid is then separated from the solid waste in a separator, and fermentation begins when the liquid is moved to a separate vessel and yeast is added. During the fermentation process, the yeast turns the maltose in the liquid into ethanol. The newly fermented and alcoholic beer is moved to a maturation tank where it is aged. After the aging period, the beer is then bottled and distributed. The process of winemaking is very similar to that of beer brewing. The grapes are first crushed and then placed into an oak or stainless steel vat. This is where the fermentation process begins. The yeast for the fermentation process comes from the yeast naturally found in the skin of the grapes. The grape juice is then aged until its specific date. It is then bottled and distributed.
The main difference between beer fermentation and wine fermentation comes from the different sugars naturally found in their ingredients. The sugar in wine making is fructose, which is naturally found in grapes. The sugar in beer brewing is maltose, which is naturally found in the malted grains, which make up the wort. The wort is a liquid extracted from mashing that contains the various sugars. Now, as we transition to our next topic, we'd like to hear a few words from our cheese man.
Well, g'day, Curd Nerds. Today we're making a taste test on Tijerina.
Another process that uses submerged cultivation is the art of cheese making. The main difference between fermentation of cheese opposed to that of wine or beer is the product. While the fermentation product of wine and beer is alcohol in the form of ethanol, the fermentation product in cheese is lactic acid. Cheese is made from milk, which contains the sugar lactose. Milk undergoes the natural souring caused by lactic acid produced from the fermentation of lactose by fermentative lactic acid bacteria. This accumulation of acid decreases the pH of the milk and causes the milk to curdle into what we know as cheese curds and a byproduct called whey.
As seen before, the glucose in lactic acid fermentation undergoes glycolysis to produce two pyruvic acid molecules, which can be deprotonated to pyruvate. The main difference between the ethanol fermentation and lactic acid fermentation is that lactate dehydrogenase reacts with the pyruvate from the pyruvic acid. This does not create the acetaldehyde intermediate or result in any carbon dioxide formation as the ethanol fermentation does. At this point, the pyruvate undergoes multiple proton transfer reactions with NADH to acquire 3 hydrogens, resulting in lactic acid. This regenerates the NAD+, as does the ethanol fermentation, which is then reused in the glycolysis process.
The second type of fermentation process is solid substrate fermentation. Solid substrate fermentation is a system where microorganisms are cultured on top of water-insoluble substrate in the presence of little or no water. The microorganisms must be supplied with sufficient oxygen, which is normally achieved through stirring of the substrate. Some of the advantages to solid substrate fermentation compared to submerged fermentation is the use of more concentrated medium, which results in less of a need for a large reactor space and a higher product yield. Solid substrate fermentation is used in the fermentation and creation of soy sauce, sake, miso, as well as industrialized enzymes and pharmaceuticals.
Fermentation has also made its way into many pharmaceuticals. New research has come to light regarding helping treat certain diseases. None of this research has definitive answers on how to cure them, but rather are approaches to help alleviate the symptoms of the host. One emerging area of study using fermentation is in osteoporosis. Osteoporosis is common in postmenopausal women and the elderly and presents itself as weakened bones, prone to breaks or fractures due to poor calcium absorption. The consumption of prebiotic fructooligosaccharides, FOS, has the potential to be a preventive method for osteoporosis. The prebiotic FOS is fermented in the gut, causing a drop in the pH of the lumen to such an extent that previously insoluble calcium phosphate will dissolve. The fermentation of FOS releases short-chain fatty acids and lactic acid, which cause the drop in pH. This dissolved calcium results in an increase in passive diffusion. Thus, it could help to treat or even potentially prevent the onset of osteoporosis. Lactic acid, a main product in fermentation in the human organism, has a wide range of uses. Hemofiltration and dialysis for kidney failure is one of the most well-known ways lactic acid helps patients. Kidney patients on standard dialysis need to have fluids replaced with something. Between 20 and 30 liters of fluid are removed from patients during 5-hour dialysis sessions, and that something is pharmaceutical-grade lactate solution. Another important group of lactate applications involves using the material directly or indirectly in pharmaceutical preparations. Topically applied lactic acid salts, particularly ammonium lactate, are used to treat severe dry skin or to mitigate the drying or irritating effects of topical corticosteroids. Lactic acid is also used to introduce certain chemicals into the human system to treat specific issues. The body is very receptive to lactic acid and its salts since it is a common process in the body. Some of the more important mineral lactates that are easily accepted are calcium lactate, which help treat osteoporosis, hypertension, as well as tooth decay; ferrous lactate, that treats anemia; magnesium lactate also helps with hypertension and muscle weakness; manganese lactate helps with glucose intolerance; and zinc lactate, which can help with certain skin disorders.